This is the cell cycle. This whole circle, cell cycle. If you look right here in yellow, you'll see mitosis. The cell spends 90% of its time in interphase, and that is where the cell is growing. Here's where it copies its DNA, and here's where it's growing some more, doing its everyday job. Whatever job that cell has, that's what's happening here. Mitosis is nuclear division. This is only a small part of the cell cycle. Some cells never undergo mitosis. Does anybody know what cells that might be? Neurons. That's a good one. So your neurons never leave G1. They're always doing their job. They don't ever divide. They don't ever copy the DNA. They don't undergo mitosis. So this entire thing is cell cycle. This right here, mitosis, is just the division of the nucleus. Okay? Before we can go any farther with DNA replication, I have to know that you know all the parts of a DNA molecule. Volunteer. I need somebody to come up and circle a nucleotide. What do you guys think?
Yeah, I have no idea. Bill's right. Probably.
There we go. Okay, you guys got that a lot faster. The rest of the class probably wouldn't have got it right, right? Okay, we need to talk about this for a minute. Remember, a nucleotide has three parts. A phosphate group, a nitrogen base, and a sugar. The name of the sugar depends on whether or not it's a deoxyribose. But take a look at this. I've got adenine, I've got sugar, I've got a phosphate group. What if I add two more phosphate groups? What would this molecule be? That's right, ATP. I added two phosphate groups. Now I have 1, 2, 3 phosphates. Adenosine triphosphate. Yeah?
Also, if you did it with guanine, it wouldn't be ATP.
If I did it with guanine, you're right. I was getting ready to go there. It's actually GTP.
Does that have a use?
Yep.
What about the C?
If I added two phosphates to the C,
It would be CTP.
it would be CTP.
So would there be TTP?
This one would be TTP. Do you remember 5' and 3'? DNA has a direction. They run opposite of each other. The two sides are called anti-parallel. This one runs this way and that one runs that way. You see they're running opposite directions? I need to show you how to count the carbons. Do you know that each one of these corners is a carbon? So we're going to count the carbons. You're going to start, the first carbon is the one that's attached to the nitrogen base. This is, for this one, the first carbon is attached to the nitrogen base. Here's my nitrogen base. So this is my first carbon, number 1. Which carbon is facing down right here? 3. 3 is my farthest point here. If I was going to attach another nucleotide, it would have to attach to 3, right? Therefore, this is my 3' end. That's my third carbon. Up here, 1, 2, 3, 4, 5. Which carbon is facing up here? 5. This is my 5' end. Okay, so my third carbon is down here.
Yeah.
So on this side, like the first carbon I would meet coming down is 5. And here, if I was coming up to touch this molecule, the first carbon I would meet is 3. So 3', 5'. That's my direction. Now watch the other side. You didn't notice this is upside down because all the letters are still right side up. The P is right side up and the C and the H are right side up, but this whole molecule is upside down. Because look, here's my 1 attached to my nitrogen base. 1. So this is the 3' side. And down here, what is this? I got 1, 2, 3, 4, 5. This is my 5' side. The one that's next to my nitrogen base. And as I counted, I moved away from the O. Okay? So I'm going to start here. This is the one that's touching my nitrogen base. That's one. And as I count, I have to move away from the O, so I've got to go this way. Okay, so that's 1, 2, 3, 4, 5. If I was going down to touch this molecule, which carbon would I touch first? The 3. That's how I can tell. And if I was coming up to touch this molecule, which one would I touch first? You're going to see when I show you the replication that you're going to have to be able to tell which side of the DNA molecule you're looking at.
Oh, is that why we're learning this?
Yes. You have to be able to tell because you can't copy in both directions. You can only copy in one direction. You can only copy from this direction up or from this direction down. You can't copy the other way. This is what I just showed you, but I had to take a little more time to show it to you. This is what it would look like. If you were counting the carbon, there's your first one. Second, third, fourth, fifth. 3' down here, 5' up there. Okay. Now, how do we put the backbone together? Remember the 3' and the 5'. Look at this bottom nucleotide. Which side is that down there? 3'. See that? 1, 2, 3. That's my 3 side down here. Okay?
So do they connect opposites?
They do connect opposites. All right. There's my 5'. There's my 3'. This molecule looks like that. So if I was building a DNA molecule, I would be adding bases to it. Nucleotides in the backbone. The backbone is referring to the side here, okay? Nucleotides are bonded together between the 3' and the 5' carbon by a phosphate group. Look, here's my 3' carbon of this nucleotide. Here's my 5' carbon of this nucleotide. How am I connecting these two nucleotides together? Right here. The
3 and 5.
Not. The 3' connects to the phosphate on the 5' end. So the 3' is connecting to the phosphate group. I'll show you. A DNA molecule has direction. We just learned that. Complementary strands run in opposite directions. There's my 5' end. Let's check and see if that's right. 1, 2, 3, 4, 5. Yes. Down here, what end should this be?
3.
1, 2, 3. Is that the carbon that's facing down? Yes.
Oh.
Good. So over here, I've got 3 at this side. See that? 1, 2, 3. And then down here, got 5. 1, 2, 3, 4, 5. Bonds. The bond between the sugar and the nitrogen base is called a glycosidic bond. That's this one. The bond between my sugar ring and my nitrogen base is a glycosidic bond. Those are strong bonds. The bond between two different nitrogen bases is called a hydrogen bond. The bond between two different nitrogen bases is called a hydrogen bond. Those bonds are weak. Hydrogen bonds are weak. Glycosidic bond, strong. This is an important bond as well. Phosphodiester bond or an ester bond. Phosphodiester.
Like,
Yes.
that injected in the
Yes.
Okay.
Okay.
Said between nitrogen.
Okay. But look. Phosphodiester bond. Look where this is. 1, 2, 3. It's between the 3' carbon and the phosphate group. So, I could ask you a test question that was something like this. What kind of bond connects nucleotides together on the same side of a DNA molecule? What kind of bond? So here's my nucleotides. 1, 2, 3, 4, okay? What kind of bonds would connect nucleotides together on the same side of a DNA molecule? And you would have to say phosphodiester bonds. Okay? What kind of bonds connects nucleotides together on the same side of a DNA molecule. Okay? Now,
So what's the type of bond between the H2C and the carbon? Is that also a phosphodiester bond? Or is that just
where?
yes.
Here?
Between 4' and 5'.
That's just a regular covalent bond, like
Okay.
from a carbon.
So you just say covalent. What does it say?
No, because look,
But isn't that connecting together?
this is one nucleotide.
Yeah.
Okay?
Okay.
This is your second nucleotide. So how are you connecting these two nucleotides together?
Okay, I was looking at this nucleotide.
Like you were thinking about these two connecting?
Yeah.
Yeah.
Is it a strong bond or a weak bond?
It's a strong bond.
And then the hydrogen bond is
Right. Now, listen to this question. What type of bond connects nucleotides together between two different strands of DNA? Hydrogen. See the difference in the question? Same side. What kind of bond connects nucleotides on the same side? I'm talking about connecting them here. If I'm talking about connecting between two different sides, I'm connecting them here. Hydrogen bonds. Guanine and cytosine have 3 hydrogen bonds. Adenine and thymine have 2. We haven't got to the complicated part yet. Still.
Before we know we're going to be doing string theory. We haven't gotten to the complicated part.
Phosphodiester bonds are between nucleotides on the same side of a DNA strand. All right. Here's your base pairing. You need to remember this. This is just straight memorization. Adenine and guanine are both double-ringed molecules. They have 2 nitrogen rings. See that? Right. Here's one. Here's the second. See that? Adenine and guanine are similar because they both have the double ring. They are both purines. A purine is the type of molecule that they are. Cytosine and thymine have single rings. Now, you can imagine that your DNA strand would be straight like this, right? Pretty much. It would not look like this. In terms of distances in the middle. Because it doesn't look like this, you can't bond a guanine and an adenine together. It would be too wide. That makes sense? And then a cytosine and a thymine in here would make your molecule look like that. So instead, we got to bond them A with T, so we got a wide one and a short one. And here we have guanine. So let me just say this in case it didn't click with you. This is not the bonding. This is the type of molecule. Adenine and guanine are both purines, but they do not bond together.
I got that question wrong on the exam.
Okay. Cytosine and thymine are both pyrimidines. They do not bond together. That's the type of molecule they are because they are one nitrogen. What do you suppose is right here at this point? A carbon. What do you suppose is right here at this point? Why are there 2 lines there?
Double bond or 4 bonds.
That's a double bond. What does it mean, double bond?
Bonds twice, back and forth.
Be more specific.
Two.
They're sharing 2 electrons. Good. I just wanted to make sure I said that in case it shows up that you understand. Here I've got a nitrogen sharing 2 electrons with a carbon. Anytime you see a point, you assume that's a carbon. All right. There's your pairing rules. A to T, apple tree, and cowgirls.
Is it U from G?
We're not talking about RNA. Hold it. Don't even think about it. Nope, not for a long time. Not for like 2 weeks. But then you're allowed to think about it. For now, no use. A will pair with U later. All right. Replication.
Go back. And then replication introduction. Now I'm good.
All right. Replication of DNA. We're almost to the complicated part. Almost.
That wasn't the complicated part?
No, that was the background information.
What was your little saying? Apple tree and what?
Cowgirls. Okay. Remember that your original strand is going to be your template for a new strand. When you copy DNA, each new set of DNA has half of the original DNA and half new DNA. So take a look here. If the blue is my original DNA strand, I split it down the middle, I break those hydrogen bonds, and half of it goes over here, half of it goes to this side. Then I copy it by filling in the remaining half. Does that make sense?
Where do you get the material from?
Ah, I'm going to show you. That's the complicated part. But before we get to that, on your assessment statements, one of them says, state that DNA replication is semi-conservative. And if you just state that and don't know what it means, it's not going to do you any good. So I want to show this to you really quickly. Semi-conservative is the top model. That's where one DNA strand here is split, and each new strand gets half of the original DNA and half new DNA. That makes sense? Semi-conservative is where the DNA strand is split in half, and each new strand gets half of the original and half new. Exactly. Conservative, which was proven to be incorrect, it would be if you kept one original strand complete and the second cell got a completely new set of DNA. So this is how they would, if they were going to do conservative, this is how they thought it might have happened, but they proved this to be wrong. Okay? The other way they thought it might have happened was dispersive, where some of it was all new and some of it was all old, and it was sort of fragmented together. That was the dispersive. Which one of these is the actual way it gets copied? Semi-conservative. Good. 50% old, 50% new. There's an experiment, Messelson-Stahl experiment is how they figured that out. When you go home and you look at this PowerPoint, you can click, I put a link here for you, to watch this YouTube video that explains this experiment. Basically, they grew nitrogen, or they grew bacteria in different types of nitrogen, and they were able to tell by how much the nitrogen weighed, whether or not they were getting all new DNA or some new DNA, and how that happened. So you can go watch that experiment later. For now, we're going to stay on replication. First step, unwind your DNA. You cannot copy DNA until you expose the nitrogen bases in the middle. To unwind it, you must use an enzyme known as helicase.
What are you going to be exposed?
The A's, T's, C's, and G's. That's the actual code of your DNA. You want to expose your nitrogen bases. Now, you don't know what's coming, but to help you, we're going to make a chart, okay? On one side, you can put enzymes. You might want to write this on your paper. And then a T-chart will be fine. And on the other side, you can put function. I'm going to blow your brain up with enzymes, and you need to know each one of them and what they do.
Right now.
First one is helicase. Now is as good a time as any. What does helicase do? Yes. Unwinds the DNA. Unzip it.
Okay.
Pull it apart. It's breaking the hydrogen bonds down the middle.
You actually pick up line with that?
Yes. I wish I was helicase so I could unzip your genes.
Oh my god.
Get it? Alright. Okay, so helicase. Unzipping the DNA. There it is. When it unzips, it creates what's called a replication fork, which is just like the spot of unwinding. Not too bad, right? Can go in both directions. Now, once your DNA is unzipped, it reacts with single-stranded binding proteins. Single-stranded binding proteins stabilize the DNA to make sure nothing happens to it while you're waiting for it to be copied, because you don't want your DNA to get damaged or to have anything bad happen.
So do you consider them an enzyme?
They are not an enzyme. No. So we're not going to put them in our T-chart. Single-stranded binding proteins to stabilize in the DNA after helicase comes through. Yes?
So, like, if something happens, that's where mutations.
Yes, we can have mutations in here, but I'm going to show you that they're extremely rare because we have a proofreading enzyme.
Okay.
We'll see that in a minute.
Is this might be where cancer comes from?
Yep.
Mutations?
Yeah, we're going to talk about that too.
Okay.
Okay. There's your replication fork. There's your single-stranded binding proteins. Here's your helicase. Everybody okay with this picture?
Wait, where's the replication fork?
That's just like where this DNA is splitting at.
So what are the proteins binding exactly?
They're binding to the DNA strands that have already been split and stabilizing them. Okay, second step for replication is to add new DNA to your strand. This is done by DNA polymerase 3. That's an enzyme. You can write it in your chart. And who asked where the new DNA? Yeah. So this guy is in charge of bringing new DNA nucleotides and attaching them.
Can you write the forward? Oh, it's right there. Never mind. We should have 3.
Okay. Here he is. And there we go. There we go. And you can imagine him moving up as he goes and attaching new nitrogen bases. Is that okay? Okay. But there's some more information you need to know about this. If you're creating a bond, it takes energy. Okay? Where is the energy coming from? And ATP and who else? And CTP. And GTP and TTP. Okay? These nucleotides are going to bring their own energy for bonding. Okay, so we got to bring our own energy to do this. It takes energy to build a molecule. We're going to need energy to build another side of our DNA molecule. That energy is going to come from this guy, ATP. You guessed it. And then we can take off a phosphate and make ADP. Or if we take off 2 phosphates, what is that? Well, it's AMP, you see it there, but what would you call that? A nucleotide. So that's what we're going to get our nucleotides from to build our DNA molecule. Of course, that's going to take energy, and we're going to use that energy to build phosphodiester bonds.
How is CTP and GTP and TTP? Where do they come from then?
We build them in our body.
Like how?
You don't want to know? No, I'm just kidding.
Do we not need to know that?
You don't need to know that.
Okay, never mind.
Okay. Okay, so here, same thing with G. See that? GTP broke off a couple phosphates, and I made a nucleotide. GMP. Here's another one. Same thing. Okay. You guys get the point? Nucleotides arrive as nucleosides, which are DNA bases with 3 phosphates. We can use these phosphate bonds to help create our phosphodiester bonds. We need energy to do that. Yes.
Is the replication done then?
Nope.
This really messes up the own.
You have to understand how it happens first. They arrive with their own energy, and they're bonded by DNA polymerase 3. So we're back to DNA polymerase 3. What is his job? Add new nitrogen bases. You don't need to write everything on this slide.
This is what just basically summary what you just told us, right?
Yes, this is exactly what we just talked about. Yes.
Are the what do we call it, like the sides of the C and the G always curved in the frames?
No, that's just so that you can see them fitting together. That's sort of like a cartoon drawing, so that you can tell A matches with T and C matches with G, but they don't really look like that in real life. All right. Now, adding bases. This is where it gets complicated. You can only add nucleotides to the 3' end of a growing DNA strand. Adding, yeah, this is part of this. I'm talking about DNA polymerase 3's job still. There's some rules, okay? You can't add DNA in both directions. We can only go in one direction. I'm going to have to teach that to you. Okay, we need a starter nucleotide in order to bond to it. We're going to talk about who that is in just a minute. So, take a look at this picture. Just listen for right now. Just listen. You can go back and watch this later. I need everybody's eyes. Guys, I've got my 3' end up here and my 5' end down here of my original DNA strand. I can only build a new molecule in this direction. Okay? But I need you to realize something. You're going to have to pick one way to memorize it and use that way every time. Because if you try to memorize both sides, you might mess yourself up. So, on my original strand, I can only add DNA from the 3' side. On my original strand, I have to start at 3 and I have to build this way. Okay? Never can I start on my 5' side and build the other direction. You can't do it, and I'll show you why in a second. You have to start on the 3' side.
So then why does your one thing says on this goes from 5' to 3'?
This is why. The new strand is running the opposite direction. It's growing from 5' to 3'. Okay? I need you to pick one side and remember that side so that you can always figure out the other side. You're either going to remember always, this is the way I remember it because it's easier for me this way, my original strand, I have to start at the 3' end of my original strand. And if I start at the 3' end of my original strand, I know that this is always going to be a 5, right? Because they're opposite. Yes. This one is going to grow this way. Yes.
They're both going down. This one's going 5 feet.
Yes. So, pick a side. Put it in your memory. I chose this side. If you want to always remember that you're building one from 5 to 3, then you can do it that way too. I choose to remember that my original strand, I always start at the 3' end of my original strand. And I'll show you why that's easier in a second when we're looking at how the enzymes are working. So, 3' to 5'. My new strand is growing from 5 to 3. It's called BYOE. Bring your own energy. Did you see that? Watch. DNA polymerase, there's my ATP. Did you see that ATP pop in? I'm going to break off these two phosphates and use that energy to create what kind of bond? Covalent what? Phosphodiester bond. And you see this phosphate? When I break this bond right here between these two and this one can attach right there to that 3' carbon. Watch. Play it again. DNA polymerase 3 brings in a nucleotide. Phosphate bond is broken. Nucleotides attached. Here's another one. Got my 3 phosphates. I'm going to break off those two. Attached to my 3' carbon. Another one. Another one. I just built a new strand. Okay? It is 5 to 3. My growing strand went from 5 to 3. My original strand is 3 to 5. Okay, this is what this looks like. So here's one DNA. I split it. Bam. What enzyme split that DNA?
Helicase?
Helicase, good.
Is it helicase or helicase?
I've always pronounced it helicase, but I've heard it pronounced helicase too. Can you guys tell me what should be up there at the top on that one? That's a 5'. And if you were going to count it, it would look like this. Starting at my nitrogen base, going away from the O. 1, 2, 3, 4, 5. 5' in. Okay? Yeah, I haven't got to the complicated part yet. Okay. You need a primer. DNA polymerase 3 cannot just attach to a DNA molecule. You have to have a primer first. Primase is called RNA primer. We're going to get to him in a minute, so don't worry about it right now. It's going to come down first, and then DNA polymerase is going to be able to add. Now, which side can I add going down? The right or the left? Which side would I be able to add and go down? What?
Right.
The right side. Because my original strand, I have to start at the 3 side on my original strand, right? So I can add here, going down. What would I have to do over there?
Go up.
I would have to go up. Because I started my 3' into my original strand. I would have to go up over here. That's a problem.
Why?
Because in my replication work,
It didn't go off.
okay, if I'm going this way, here's my 3' into my original strand. I can add going this way, right? So as this opens, I can just keep going and going and going and going. But up here, it's running the other direction. Okay, I can't add going this way. I have to add going this way. So I have to add some, and then as it opens up more, I have to come back in, I have to add it again, and add it again. I gotta work in the other direction. That's the complicated part. I will show you. Okay. Add these, good to go, right? That side went from my 3' to my 5' and I added my new. Perfect. Now watch this. If I try to do the same thing on the other side, look at the orientation of that molecule. Now watch. I added another one. Where are my phosphate groups?
On the sides.
Okay, but they're down here, right? I'm going to break this bond,
Oh,
and what am I going to attach it to?
attach what to?
Exactly, this phosphate. It needs to attach to the next nucleotide, right?
Understood the problem.
You see the problem?
See, look up there. See, look, there's no, because I broke the bond before I formed with the T.
Okay, I need these 3 phosphates to be right here in order to attach these together. And they're not. They're in the wrong direction. That's why you can't build it this way. You can't do it because I broke off the phosphates. Now what am I attaching it to? There's nothing there.
Can you go back to the first one?
Up here?
And use the red one? Like building
Okay, building this one. Watch. See where my phosphates are? They're facing the nucleotide before it. When they break off, they attach to that nucleotide. They break off and they attach. Break off, attaches the nucleotide. Breaks off, attaches the nucleotide. When I get to this side, if I try to go this direction, see that? There's no nucleotide there for the phosphate to attach to. Right. No, you can't attach them on that side. You would have had to have phosphate, and if you kept doing it, you would keep running into that problem. Okay? But here's how we fix it. We're going to start down here and we're going to go up. Like you guys told me originally, I needed to go up on that side, right? So watch. There it is. Now look, I'm going up and it's there. I'm going up and it's there. It works to go up on that side. It works to go down on this side. Now I gotta start down farther and work up again. Then I gotta go down and work up again. Now I have a problem, right? Don't worry.
Oh, so this is the whole complicated part?
I'm almost there.
Oh.
Yeah. Ligase joins fragments. We're going to go ahead and call them what they are. Okazaki
That's the complicated part.
fragments.
It's like Asian.
Okay.
Name after somebody?
I had to start down here and go up. As my replication fork is opening, I'm going to keep having that problem. I'm going to have to keep go back and go, and then go back and go, and then go back and go, and then go back and go.
I can just start from there and start from the very bottom.
What do you mean, start from the very bottom?
When you're not being gassed.
If you waited until you completely unwound it,
Yeah.
Doesn't it like never completely unwind?
you can't do that. You have to go. You have to do it as you go, or your DNA will be exposed. So let's say.
Oh, because then your DNA will be damaged and there will be mutations. So you
Yeah.
have to start it.
So you're unwinding it,
Because it's
okay? You're unwinding it, and this one's copying this direction. Copy, 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 copy. And as you unwind it and unwind it, it just keeps copying, 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 copying straight. No problem. But this one is running in the other direction. So as you unwind it, you can copy this section. But then when you unwind it more, you've got to go then and copy this section. And you got to copy this section. You get all these fragments.
Okay, so the bottom is like the right side.
Yes.
Okay,
And the top right here is like this.
so I'm not understanding.
I'll show you after you see the animation, and we'll see if you. I think the animation will help you. It's hard to draw it on the board. So for now, we're doing it in sections. And if you don't quite get why they're fragments, right now, don't panic. In the animation, you'll see, and it'll make sense why you'll have fragments.
I have a question.
Okay.
So, is it always going to be with the old side? It's always going to go from down to up? And you said it's always going to go up to down? Or?
No, the old side always starts replicating on the 3'. I could have switched this DNA molecule around, and I could have had my 3' over there. You have to pay attention to this, and you can start going down from here. And your new strand is always going to be the 5 to 3. Okay, here's what it looks like. There's my what enzyme? Helicase, good. What are those black things?
The polymer.
Single-stranded binding proteins. Good. Those guys are keeping the DNA stabilized once it's been opened. Now, here's my 3' and 5'. If I tell you my leading strand is the one that can copy straight forward, no fragments. Which one of these would be my leading strand? The top or the bottom?
Because it goes from 3 to 5. No, the bottom, the 3 opens. I have no idea what you just said.
Did you say the bottom? You both said the bottom, right?
Yeah.
No, she said bottom. So which one is the leading strand? Look at that picture. Could be in lazy brains. Leading strand is the one that can copy without going back and doing any fragments. Alright, Monica, you had it right.
I know.
If I was going to copy this as it's opening, I can start here. This is my original strand. I can just copy straight down, right? Because I can copy from 3 to 5, no problem. And that's going to continue to open. I'm just going to continue to copy. You guys had it right. Up here, I can only copy from 3 to 5. So as this opened, I copied this direction. And then when it opened some more, I copied more of it. And then when it continues to open more, I'm going to copy more. But I got to wait till it opens some and then copy backwards. Okay with that? There he is. That's good.
So you can't just wait for the bottom to be open?
You can't wait. You can't open. You never open up like an entire. You're talking about billions of bases. And you can have multiple helicases on one chromosome. You can have one chromosome, and you can have replication forks in different areas.
Are the fragments all the same size?
No, they don't have to be. All right, take a look at this. Can't copy that way. Got to copy this way, right? There's your Okazaki fragments.
Why are they called Okazaki?
Ligase. What's ligase's job?
So it's Okazaki fragment.
There you go. There's your leading strand.
I think I'll ever forget that.
Lagging strand.
You need to get this thing checked.
Okay. Lagging strand has Okazaki fragments that are joined by ligase, sort of like a spot welder. The bubble is like the whole part that you see. So check this out. If I have 2 molecules, if I have 2 helicases working in opposite directions, if I'm going this way to the left, which one is my leading strand? The bottom. Check. If I'm going this way to the right, which one is my leading strand? The top. Because if I'm going this way, 3 to 5 is up here, so I can copy that way straight, no problem. See that?
Is the leading strand always going to be the oldest strand? Basically.
Is it always going to be the way?
The older strand. Because it goes from 3 to 5.
No. The leading strand depends on which direction you're copying. If I'm copying this way, if I'm opening this up this way, I can only copy from 3 to 5. So I'm going this way. This is my leading strand. But if I'm opening it this way, this one is not 3 to 5 anymore. It's 5 to 3. You see that? So if I'm going this way, 3 to 5 is on top. This is my leading strand on top. So it depends.
Wouldn't it be,
Wouldn't it be what?
wouldn't the leading strand and the lagging strand switch in the middle of the fork?
Right, but so on a test, direction matters, okay? On a test, I won't ever show you this picture like this and ask you which one is the leading and lagging strand because it depends on which direction you're going.
Well, from there it looks like neither of them are because one would be, because you're copying, if you're both, I don't know.
If you're copying on, yeah, technically in the middle, this would become your leading,
Yeah.
and this would be your leading, and this would be your
Yeah,
lagging.
that's what I'm asking. Like, well, if it asked you for this picture, what would you say? Because it looks like they switch down the middle with his leading.
It will
and his
ask
lagging.
you. You'll never see this picture without any other direction.
Oh.
It'll have one arrow or the other,
Okay.
okay?
Okay.
If I showed you both, I couldn't ask you which one is the leading and lagging strand because it depends on which direction you're going.
So if you just showed the right arrow, say the top one is the leading strand.
Right.
Now I understand why you think it's complicated. Wait, this isn't the complicated part?
This is the complicated part. The fragments is really hard to
I
understand.
thought you just wanted to like
I know, that's what I'm saying.
This is your bubble. Okay, this kind of shows why it's just drawing on the board.
Oh,
Okay.
That's gross,
it's like a circle.
Or a potato.
Yeah. What? Look at a circle.
What's the circle?
That's a Chandler
Well, it's
circle.
because like,
It looks like a car.
See, it's all going in one
oh my
direction.
god, it does.
If I change
the corn pig.
the 3 and the 5s.
Oh, it does look like a corn pig. Just for pig.
Yeah, let's see.
Okay. Starting DNA synthesis. I did mention that you cannot use DNA polymerase 3 on its own. You can't just attach it to a replication fork and have it start working. Something else has to go down first, okay? So, we call that RNA primase.
Ew. That color. I like this one.
I don't like it with the red.
I don't like the pink.
Yeah, that's just not my color.
Okay, RNA primase's job is to allow DNA polymerase 3 to be able to attach.
How does it do that?
DNA polymerase 3 can attach to an RNA primase. It cannot attach straight to the DNA strand.
Why?
That enzyme does that. That's all you need.
So it finds it.
Exactly. Getting it ready. Kind of like sanding something before you stain it. The stain won't stick until you sand it.
Or you put like
Primer on it. Primer before you paint. Once you got your RNA primase, then your DNA polymerase 3 can attach and start doing its job. There's one small problem. DNA polymerase 3 on the lagging strand has the same problem. He can't attach until there's an RNA primase. So you have all these RNA primases. Do you see all the red squares? Those are all places where this had to go first before DNA polymerase could attach. So we've got all these segments of RNA primase. So we need somebody to go through and replace the RNA primase with DNA.
oh.
Don't worry.
Replace the squares with the circles.
No. We need to replace these red squares with blue.
Oh, with blue.
Okay? That's going to be DNA.
So RNA primase puts RNA there
Yes.
in order to start DNA polymerase 3. And then to fix that, it uses DNA polymerase 1, and then
Take out the
ligase,
RNA primase.
and then ligase.
Links the Okazaki fragments together.
Okay, makes sense.
Alright, DNA polymerase 1 replaces RNA primase, RNA primer with DNA nucleotides. With DNA. We don't want RNA in our new DNA strand, or wouldn't be a copy. So that's what he does. There he goes. Ooh, did you see that? Watch. Ah. Ooh. Now we've got 2 DNA strands.
Looks like a donut, and then he eats all the red.
Does anybody see the problem?
Where does it go? Because there's 5. The ligase.
Ah.
5.
Ligase joined all Miyazaki fragments. He's good.
No.
Allie, what's my problem?
There's a missing chunk of DNA.
I have a missing chunk of DNA.
So that was just laziness.
No, I have a missing chunk of DNA where there once was my RNA primase originally. DNA polymerase can only work from 3 to 5. I can't run 5 to 3 right here. I cannot add DNA in this direction. I can't.
Why is there a blank spot there?
Look.
Go back. I had a question a while ago, but you switched it. How come the RNA is behind the other stuff if it's supposed to be priming? That doesn't make any sense.
What do you mean?
Go back to the last slide, that one. How come RNA is behind the DNA column? Wherever.
Here?
Yeah.
It came down first, and then the DNA attached and was going.
Oh, so it does come down first because it's behind.
Yes, first, and then it's running this way.
Okay. Throw about the brain down. Yeah, we need a brain break.
You can handle it. You're almost done. It can't just turn blue because blue would mean that I added DNA there. This is the problem. You lost some of your DNA during replication. Every time your cell divides, yes, you lost DNA. But look, don't worry.
Where does the DNA go?
You weren't able to copy that part because originally there was an RNA primer there.
But he moved.
Isn't DNA polymer?
Yeah, then it's too late to copy it.
It's too late to copy it now. It's in the wrong direction.
And there's already the other part of it.
This is our...
Wait, I want Mr. Lynch to explain it.
Okay, you can't copy that. It's going the wrong direction now to be able to add polymerase. But don't worry. You have something to help you with that. Your body is incredible. And you have extra DNA on the end of each of your chromosomes, of your genes. They're called telomeres. Have you heard that word before?
No. So we are born with a whole bunch of extra DNA just for this purpose?
Yep.
So when you get old, that's why you start deteriorating?
Mm-hmm. That's why your cells can only divide a certain number of times before they're no good. Okay? Now,
That's why you die.
check this out. You have a loss of bases at, you have a loss of bases at the 5' end in every replication, okay? Chromosomes get shorter with each replication. That's why there is a limit to the number of cell divisions. You have something, though, called a telomere. A telomere is a repeating, non-coding sequence at the end of chromosomes. It's a protective cap so that when you copy DNA and you lose that little segment because of your RNA primase, which, by the way, is about 10 nucleotides, you have this protective cap so that when you lose that, you're not actually losing any of your genetic information. You're not losing anything that codes for anything. This is extra DNA, non-coding segments. We are going to have a way to fix that. With an enzyme, don't worry, this is the last one. Polymerase adds DNA. We can no longer use that segment. What type of cells would you see a high amount of telomerase in? Okay, cells that do a lot of division. What type of cells, even more than skin cells, would you see? Huh? No? Cancer cells and also stem cells. you would see a high amount of this enzyme. All right. Here's what you should know from today.
We're going to review this again, right? That's what we're going to do.
No, we have to do some stuff with it. This is not it. It's like learning cellular respiration. It's really hard at first, and then you practice it for a couple of days, and then you get it down. Yes, yeah. Okay, so this is what you should know. There is a direction for replication. Your original strand, the direction is 3' to 5'. On your growing strand, the direction is 5' to 3'. Enzyme that unzips DNA is called helicase. SSB are single-stranded binding proteins. Keep the DNA stabilized. Primase is added so that DNA polymerase III is able to attach and add new nucleotides. Okazaki fragments are created because the lagging strand runs in the opposite direction and must therefore be copied in sections. Ligase binds Okazaki fragments together. DNA polymerase I will replace RNA primases that were laid down originally. This is your leading strand, and that's your lagging strand. By this time next week, you should be able to draw and explain this whole picture.
So you don't even telomerase?
Yeah, you need to know that one. It's just not on this page.
Polymerase 2.
Oh, 2? We haven't talked about 2 yet. Later, my friend. All right. A couple of little things. DNA polymerase III does 1,000 bases a second. 1,000 bases a second. That's how fast this works. Now, you have a lot of DNA, so it would take your cell forever to copy it all if you didn't have DNA polymerase III to work that quickly. Polymerase I can do about 20 bases a second, which is still pretty quick. For editing, repair, removal, let's see, 1,000 bases per second. You do have a lot of typos. Those are fixed by DNA polymerase I. Oh, you asked about that, right? Mutations? Okay. Originally, you have 1 in 10,000 bases as a mutation. But after DNA polymerase I goes back through and corrects it, it's 1 in 100 million.
Probably corrected on the leading strand if it can't replace.
It does actually slide along it. When it's replacing the RNA primase, even on the leading strand, it'll slide along and correct any errors.
Then why can't it replace the telomere?
Good question. Why don't you look that up and let me know? See if you can find that out. You might have to write it down. It takes E. coli 1 hour to copy 5 million base pairs in its single chromosome. It divides to form 2 identical daughter cells. Human cells can copy 6 billion bases and divide into daughter cells in only a few hours. 6 billion bases.
What's the difference between daughter cells and son cells?
There's no such thing as a son cell. They're all called daughter cells.
Okay.
All right.